Good afternoon, everyone. So if everybody can come in front, so I think we would have, because I think industry is the one which is present predominantly here. So I think this we would make as I had a presentation in Miami last week. So the first opening session was a unplugged session, which was predominantly with all the industry present there. So I think industry is something which is quite helpful, and I think they have sometimes more answers than we have. As far as, so I see, industry representation from biosimilars here. So means the reason behind organizing this instruction course is that, if you look at last means 3-4 years, if you look at some of the leading molecules like abicipar, means I'm not sure if you are quite familiar with abicipar. Abicipar was a molecule from Allergan. So Allergan means it was quite a promising molecule with a kind of platform where it's like a thread. It's like a thread chain where you can add any kind of drug. So it means it was such a wonderful platform, and we were all hoping that that molecule would come and make real big difference. But unfortunately, if you look at the incidence of inflammation was approximately 18%. So 18% nobody would tolerate, because you have molecules like ranibizumab, which has just set a standard to a different level altogether. Means so 18% was the immunogenicity, and although means not then FDA gave some instructions and company could identify it, because there were a lot of E. coli material which was present, so they filtered E. coli material. In spite of filtering that E. coli material, means incidence of immunogenicity could not be brought down less than 8%. So 8% is too high. So unfortunately means that molecules became dead in spite of a wonderful molecule. So immunogenicity means plays a huge role as far as newer molecules are concerned. So that's what we are going to talk about here, that how immunogenicity has impacted the entire industry, anti-VEGF industry per se. And now means India leads in biosimilars. I was in Miami, so I had 3-4 sessions on biosimilars. So West has no idea. West is still skeptical. So it means we need to talk about this and probably take our experience to the global level, that how means we should be confident about these molecules and development process, and why the development process is curtailed down. So means we would talk about all those aspects. Even brolucizumab, if you see, means I'll talk about brolucizumab, such a wonderful molecule, but means the kind of success that it could have had, it could not achieve. The kind of drying that you see, I will show you some of the examples, but immunogenicity has a role at this point. Gene therapy, entire gene therapy is on the back seat because of immunogenicity. So it's an important topic to talk about. So we'll start. Nilesh, very young guy, means he was my student. He did DNB with means we're both together. I learned a lot of things from him. So he means as of now he has established himself. He's doing private practice. Means I have learned so many concepts from him. So means he's quite accomplished and established boy. And he would talk about that how I per se has a lot of means it's a separate organ. Eye is a separate organ which is supposed to be immune privileged. But in spite of that, all these therapies which are intravitreal still faces challenge of immunogenicity. So he would talk about the basics, that what kind of immune privilege that eye has. Nilesh, over to you.
Thank you so much, sir. And sir is always kind with his words, his praises, and I don't deserve much, because everything I am today, I am because of him. So a big thanks to AIOC Scientific Committee and Ashish sir for giving me this opportunity. I'm going to talk about, we will talk about how to understand immunogenicity in the eye. So these are my financial disclosures. So we already know that immune response is a sequence of molecular and cellular events intended to get rid of the host of a threat, offending pathogenic organism, toxic substance, cellular debris, or neoplastic cell, which may be either innate, that is from inside the eye or inside the body, or adaptive. Inflammatory response theoretically is a clinically evident immune response. So understanding the ocular immune privilege, which means the loss of immune response in the eye, has developed over time. First described in 1950, Medawa described immunologic ignorance to be based on passive mechanisms such as blood-ocular barrier and the lack of lymphatics. Kaplan and Sterling in 78 described immunization through anterior chamber created systemic deviant immune reaction. That is, when any pathogen or any allergen from the eye goes into the system and systemic circulation, there is no antibodies formed against it. That is the immune deviation that happens. Current understanding says that immune privilege is an anatomical segregation and a complex interplay of other active mechanisms, which we will see. These active components include immunological ignorance, peripheral tolerance to eye-derived antigens, and the immunosuppressive microenvironment in the eye itself. So the passive factors are blood-ocular barrier, deficient lymphatic drainage, intravenous drainage of aqueous humor, so it directly goes into the vein, reduced intraocular expression of the major histiocyte complex, both class 1 and class 2, and the fewer antigen presenting cell, be it from iris to retina, anywhere. The active components include cell surface expression of different ligands and soluble factors in aqueous humor such as TGF factor beta, alpha MSH, VIP, interleukins, and soluble CD95 ligands, and thrombospondin. So innate immunity is an antigen-independent reaction having immediate maximal response, which is not antigen-specific, and there is no immunological memory. These are derived from lactoferrin, lysozymes, defensins, complement systems, mannose binding lectins, all the pathological mechanism that is possible there. In contrast, adaptive immunity requires antigen, which has to be recognized by the immune system. So it is very specific. These antigens are recognized by the presence of epitopes. That is very important for our understanding of anti-VEGFs, which is then processed by the system, and then there is an immune reaction which can be delayed too. This reaction is antigen specific, and the immune system creates a memory to the particular antigen. So that is one very important point to be noted, that there is a memory to be created, and it is rarely first type reaction. The immune response of adaptive immunity has been described by 4 mechanisms. Type 1, that is the anaphylaxis reaction, that is IgE-mediated immediate reaction, while type 2 reaction is performed by cytotoxic T cells via IgG or IgM antibodies and happens within few hours to few days. Type 3 and type 4 we will see. The immune response can also be divided based on humoral, that is the antibody-mediated, and cellular, that is the cell-mediated. So humoral response is via antibodies produced by B cells targeting extracellular microbes and toxins, which will be important for us to understand, interacting via B cell receptor, which can also recognize unprocessed antigens. In contrast, cellular response is very specific to, via T cells responding to intracellular microbes, their products, and tumor cells. So all the reactions that can happen to the molecule itself will happen through humoral response, while anything happening to the debris in the preparation, that will happen through the cellular response. So type 3 hypersensitivity reaction is a humoral response involving IgG antibodies, which forms a chain with the antigen and get deposited in the tissue. So this is very important, what we have proposed for brolucizumab also, and sir will be talking about it, that there is a formation of a chain that gets deposited into the tissue, and over subsequent time, if the deposition gets more, it can lead to occlusive vasculitis, which was a new phenomenon with brolucizumab. So these deposits attract complement, leading to neutrophil discharge, and the complete process may take few weeks to be clinically evident, as again seen in brolucizumab. In contrast, type 4 reaction, which we assume brolucizumab is having, is just because it was delayed. It was presenting 4 to 8 weeks, but it is, as we know, inflammatory response is the clinically evident immune response, so it takes time to get clinically evident. Cellular response, I think everybody knows, it is first there is an exposure to an antigen, there is a prior exposure that is required, and then subsequently the second reaction will be like an anaphylactic reaction when suddenly the patient will develop reaction. So this is one hypothesis that also holds value for brolucizumab, because it happens with second dose mostly, not with the first dose. And again, there is preformed antibodies, which again we'll talk about because it is a smaller protein. So it has preformed antibodies against it also. So now antigens can leave the eye via 4 gates only, which are lymph vessels of conjunctiva, anterior chamber angle, and the Slem's canal there, retinous venous circulation, and the choriocapillaries. The lymph node of ocular circulation is the spleen. There is no lymph node in neck or anywhere. It is the spleen. So any antigen presenting cell initiate T cell maturation should reach the spleen. The peripheral tolerance to the antigen happens in the spleen. So let's understand some part of sources of immunogenicity. I think I have told some part, and then sir will continue with his talk on brolucizumab. First thing to understand is why does not all injected biologics, which essentially are foreign or non-self, induce an immune reaction? This is done by making the antibodies more humanized. That is, essentially leaving the epitope as it is and changing the rest of the things to become more human, so the body doesn't consider it as a foreign antigen. The humanized antibodies are tolerated by our immune system through the self-tolerance pathway. The molecule, however humanized, still are all immunogenic. The vehicle itself can carry cellular debris, degraded protein fragments due to oxidation, deamidation, glycosylation, and aggregation of such protein itself can cause debris. And they also carry endotoxins from the cell line. The injection technique itself can cause a reaction, as we have also published in an article that the silicon lining of the injection itself can induce an immunogenic reaction. The details of each component will be dealt by sir again in his talk. The immune reaction can be enhanced by localization and prolonged presence of the biologic in an increased concentration in the proximity of antigen presenting cell, and such factor may lead to inflammation. So if we are giving more amount of antigen very near to the antigen presenting cell, like in brolucizumab, we are having a very high molar dose, so that can also lead to inflammation. The antigenic site on the antibodies have been termed as idiotopes. This is a new concept that we are trying to push. It is not a new concept, but we are trying to push into the biologics segment. That is a formation of anti-drug antibodies. Anti-drug antibodies essentially mean when we are injecting a biologic, there is a small amount of immune reaction, and with subsequent injections, this amount of antibodies, ADS, keep increasing, and that may lead, that can be one of the factors that leads to either tachyphylaxis or intolerance or failure of the therapy in subsequent stages. So the AD are produced via T cells dependent pathway if the biologic is processed by antigen presenting cell, while few can directly interact with B cells to convert the B cells to immunoglobulin G producing plasma cells. These IgG ADAs development can be triggered by an exogenous human protein or by the failure of immune tolerance to the self antigen that can lead to overt immune reaction. The ADAs can be either binding or neutralizing. They can either bind to the drug and make them deposited in the cellular debris, or they can neutralize it and the drug won't have any effect. The new immunological debate that has been brought in focus due to retinal occlusive vasculitis, which has been reported following brolucizumab, while there have been many theories put forward about the pathway, including our group, the only clinico-pathological correlation was done from a vitreous sample taken from a patient which majorly showed CD3 positive cells, which are immature T cells. There also has been some CD4 and CD8 cells and also rare B cells. So it looks like there is a complex interplay that is happening of type 3 and type 4 reaction that is leading to that peculiar reaction, that is the occlusive vasculitis, which was never seen before with the anti-VEGF agent. So over to sir. Thank you, sir, for this opportunity, and we will continue, sir. Thank you.
Thank you, Nilesh, for giving the overview of the immunogenicity, how it works. So predominantly means all the attendees, it might have been quite a complex talk, right? Because immunogenicity is complex. So means because we have time, so I probably, because we have industry leaders here, so probably means Manish is here. So means Manish is somebody, who has seen Razumab journey from the beginning, and they had some issues in the beginning. Means Manish, I would like to have your comment on the endotoxin part, which we dealt with, I think, second. Means if you can come on the mic.
Can we have the mic there, please? Oh, thank you.
Again, when it's come to endotoxin, it's one of the most important parameter for any biologics. And there are certain permissible limits. Like if we talk about FDA limit is different, ISO limit is different. So like, especially what we have found that in our internal studies, at any point of time, if endotoxin is more than 0.2, it might lead to inflammation, which is very much aligned with FDA. Like there's it's not that you can say that biosimilar. Even for any product which is in being used in ophthalmology, there is specific limits. Like for IOLs, there are a limit. For viscoelastic, there are a limit. For you can say that dyes, there are a specific limit. And the limit for ISO, any limit which is being decided by ISO and FDA, these are the different limits. And the limits which is being given by the FDA, it is more tightened limit. So any product which is in much more aligned towards the FDA limit, of course the chances of inflammation is going to be subsided. And at the same time, the most important factor is that even at that limit also, the chances of inflammation might exist. The reason behind it, every patient is different. Every patient has got a different immunogenic, you can say that, response to the drug. Immunity is different. So like that is also important factor. It is like that we can't say that even at that range, the product is 100% reaction free, ADR free. We can't say.
So I agree. Means this is a very important point, because immunogenicity is all about, how, means it does not just depend on, how the drug is structured, but it also depends on, how your body. Oh, Dr. Mangat is there. Sir, would you mind please coming in? Sir, please. Because we have missed Dr. Vishali. She had to go for, sir, please come on, sir. Please, I request, sir, please. It's an honor, it's an honor to have Dr. Mangat, sir, on, with us. He's very kind that he is in the session. He has been a teacher for all of us. I think he does not need introduction at all. Yes, sir. Honor to have you, sir. So as I was mentioning, immunogenicity is something that you cannot just control by controlling all the parameters that you have while manufacturing, because it's your body, body responds to a few things quite differently. So I think we have leaders from Lupin also. I'm not sure whether from Reliance. So Lupin means now your molecule is just approved just now. So Rennie Eyes. So what kind of immunogenicity tightening was done during the phase 3 for that? Dr. Natarajan, please. So it's good to have both of them with us. It's so kind from both of them. Yeah. Thank you, sir. Thank you for joining. So if from Lupin, if we can ask, because Dr. Vishali was not there, and I've just made it like Miami said, that introduction session that we were there. Yes. It's like industry session with because everybody wanted that kind of. So we both were there. Dr. Natarajan and me were there. So in the opening session, that was the kind of unplugged session. The industry was with us, and we always feel with all these molecules. We learn so much from the industry also. So Lupin molecule is just going to be launched. So we would like to have some word, what kind of immunogenicity testing was done in phase 3, if you can comment on.
Basically, sir, we have done the overall phase 3 clinical trial. That is a double-blind phase 3 clinical trial, and which is done on 202 patients. In that 101 patient that we have taken for our ranibizumab, Lupin's ranibizumab, and 101 patient was on an innovator's branch. So in terms of the overall immunogenicity levels, so we are almost equivalent to that innovator brand, sir.
Do you have any percentage to quote, or?
So there is only one patient which has been, because in the overall there are around 88 patients. So in that there is only one patient which has been come out with this, there is an immunogenicity reaction which has happened, but overall it was not.
So last time, what we were discussing before the conference, We should ask them to check for the ADAs also, because
Yeah,
it
I
is a
think
very
I-
minuscule percentage that they check for ADAs.
Okay.
So we just have a sincere request for future development of product that
So
ADAs
I think,
should be.
so means ADA, I think in all these international, we are part of some of the EM and FDA trials that ADA has become a very important tool. So I'll take one comment from Dr. Dogra sir. Means in terms of now we have approximately 3 biosimilars. One is from lupin, means which would probably be coming in. One you have Resumab and one you have from Reliance. So means at present, means do you see them from a different eye, or means what is your comment on that?
I think the way we, right from beginning, the way we worked, I think it's known to everybody. We are very careful. So that was the story even with Razumab. I think we started using it very late. And now once it is being used, I think it is used really left and right, because you develop that kind of a confidence after all those things happening. So we do get a lot of this thing from Reliance people that we should be using it. Nothing against it, the same thing. We want to look at how it is doing. Has it been used in more number of people? Because the question remains always with any biosimilar, if we have reaction or some problem, I think it has to be reported and it should come in the public domain. And that's how we build the confidence. So this thing for the lupin also, or any of the biosimilar, is same, that let's have a real good data down the line, or your users should also be very sincere, not that they should be. So that's where I think the bottom line for us is, and that's how at this time perhaps this Razumab is being used more and more, because the confidence building was because of the numbers which were reported across from various centers.
Very well said.
Yes.
I think, sir, I completely agree. Dr. Natarajan, you have any point?
I think I agree with Dr. Mangat, and same problem I had. And my patients could afford. They say, "Oh, why should I use the drug which is not internationally available? Why can't?" So the problem was the same. Well, I think the beginning we all shared. So I think that's what we had a discussion in Fort Lauderdale also. Like when you have a standard drug, when you have a biosimilar, and what's the difference in the
True.
drug? And I think you also explained there the Avastin. The biosimilar Avastin is not a offline drug, and it's a
It's
primary.
the innovator, it's
Yeah.
no, it's not right. So probably means with sir's comments and overall means it's not about just biosimilar. It's about any molecule which just which comes new, that we always take it slow, because that's what the lessons that we have learned, and just take it slow, and probably once you develop confidence, for example, the kind of confidence Resumab has given now, if you see the data from VRSI 2018 to 20, initially there were hiccups, but from 2018 VRSI data says that kind of usage it has just picked up. That's what means when me and Dr. Natarajan were there, means now US FDA has approved their first biosimilar in US, Canada and UK, and 27 European countries. So we were proud to share our experience, that how our journey was, because there is still a lot of skepticism in overseas, that how they would place this drug, because means a lot of dynamics. It's a regional dynamics means predominantly. So means whosoever player is coming in, we have opened a group which is called Bio User Group. Bio User Group is kind of real world data that we would be collecting from Japan, because Japan also from January they started their first biosimilar ranibizumab started using from Senju Pharma. So overall from the West also, this is the feedback that we have received. More and more real world evidences that we have, probably that's where the confidence building happens. So I think we would again discuss it, and I would just have my talk finished.
So I'll just introduce, sir. I think most of us know, sir. Dr. Ashish Sharma is heading the medical retina department at Lotus Eye Institute, and he has around 100 publications based on anti-VEGF. So it is a massive number and a massive experience that he has. And over to sir for the.
So the topic of IC was, immunogenicity, whether it is, it's kind of a hurdle for us in further progression. So that's what, during introduction I said, we had wonderful molecules like abicipar. Abicipar was a wonderful molecule, means could not get approval, means after that phase 3 in spite of a lot of measures being taken. So brolucizumab, I means always say excellent drying, excellent efficacy, but just because of, incidences of inflammation, it's not taking off the way it could have just taken off. So means I would talk about peculiarly ocular vasculitis, that why this has become the talk of the town, and why this is important. So means we have been trying to understand biologics, biosimilars, and all these molecules since last 4 to 5 years extensively, and thank you, Nilesh. So we have been trying to understand these molecules since last 4 to 5 years, and this is our contribution in the form of various publications. I would definitely like to mention this group, which has been very, very helpful since we have started understanding and putting it on paper in the scientific forum. So means a big thanks to all of them. And as far as my financial disclosures are concerned, these are my disclosures. So why occlusive vasculitis was highlighted? That's the first question that I should ask. Because it was never observed as an adverse event with existing anti-VEGFs in all the trials, if you see from the beginning. And other thing is, it was sight-threatening. So that's why it was highlighted. And if you look at the day of celebration in 2019, 8th October, when the drug was launched, after that few months, there was alert on safety from ASRS with 14 cases of vasculitis and 11 cases of occlusive retinal vasculitis. So then Novartis was responsible, and they had a SRC. SRC, what it did was, the question was that why it was not picked up during the Hawk and Harrier. So then they went back and they checked all the cases carefully, which were not reported as occlusive vasculitis, which were reported as just the intraocular inflammation, just to check whether in that cloud of intraocular inflammation, we had some kind of vasculitis there. So after studying that data, it was revealed that incidence of retinal means vasculitis and retinal vascular occlusion was higher than the trial. It was approximately 2.1% compared to the trial, which was 1%. So there was a disconnect. So then Novartis updated their label. For example, if you look at their label information in 2019-10, then retinal vasculitis was not mentioned. But if you look at the data and label in 2020, it was included after the SRC report. So this was the paper that we published, that there was definitely a disconnect from the trial to the real world. And we cited some of the reasons that why the disconnect was. Now, means I would just straight away come to some of the representative cases for clinicians or anybody who is there who just want to see. Look at these cases, the kind of whitening because of the occlusion, retinal whitening. But if you look at the OCT, right from the B, D, and F images, look at the efficacy, just brilliant. Efficacy was so brilliant, but occlusion was something which was worrisome. Again, if you look at the FFA, in this case, multiple occlusions, dye is not going through further. And similarly, in all these cases, if you see, again, occlusion, arrows are pointing out, FFA showing the dye is not going further. But if you look at the OCT, such a drying, that kind of impact we have never seen with any kind of molecule. Again, this case, again, FFA shows a lot of occlusions at multiple sites. So these are the cases were from my very good friend Caroline Bamal. So if you look at the structure is quite different. You have power packed, 22 times concentration compared to the other drugs. So probably that was kind of a double-edged, means you have more molecules to give you more efficacy, but means I was probably not that tolerant towards that many molecules. Question was retinal vasculitis and retinal vascular occlusion. Why? Eye is an immune privileged site. So why do you have this vasculitis and vascular occlusion coming in? Mystery has not yet solved. Still it's a mystery. So what we did was, because we have been working with biosimilars and biologics, trying to understand, means when we had few events with the Razumab, we have been publishing since that time and trying to understand from basic science that what happens. So this is something like, when you open an old wine bottle, that is more sweeter. So this is the answer that we found, and if you look at the paper which was published in 2019, way before when brolucizumab was launched, we proposed that ADA could be something which need to be looked upon, anti-drug antibodies. And after that, what happened? We proposed this hypothesis in this paper that type 3 is something which could be responsible for this vascular occlusion, and possible ADA testing before occlusion might be a way to go. And the same thing we proposed to the designer of the molecule, Doug Saur. So Doug Saur, means in a meeting I discussed with him, probably pre-injection ADAs were too high. He said, Ashish, I agree with you, but the problem is, means they were all non-specific. They were all non-specific segments from where the ADA was generated. And to get the direct correlation and to establish ADA testing, pre-injection is a difficult task. If I know that, in every case, ADA is high, induced ADA is high, then inflammation is happening, then probably, I can push it for. But again, that direct correlation was not coming in. So that is why ADA testing is restricted still till the clinical trials. It has not yet gone into, pre-injection phase. So this was the paper which was contradictory to our hypothesis from a very good friend, means Thomas Albini, who works in uveitis at Baskin Palmer Institute. So what they did was, in one case which had retinal vasculitis, they took a sample from vitreous. And in vitreous, they found CD4 and all the markers which belongs to the type 4 reaction. So they proposed that probably the type 4 is the one which could be responsible. Then this is an editorial that we published. We re-emphasized our stance and gave a reason that, all right, but means we really cannot believe that it's just the vitreous that you can find an answer. If you really want to see, because it's the phenomena which is happening in the vessel wall. So you really need to look at the vessel wall if you really want an answer, biopsy of the vessel wall. So we are still, means sticking to our hypothesis as probably type 3. But we could be very well wrong in that. Now, if you look at the Merlin data, which came after 1 year, which showed that after monthly injection, brolucizumab inflammation rate was approximately 9.3%, which was pretty high. So after that, after Merlin data, companies stopped all the trials which were giving monthly injections after initial loading doses. Now, if you look at this, and if you look at our paper, we proposed it way back when we proposed our hypothesis and the mitigation plan. We proposed it in November 2020 that monthly injection is not the right way for brolucizumab. So that's what happened over when later Merlin results came out. So now what? Now the question is, now do I use this drug or no? I use this drug, but it is not my first line drug. It has a great efficacy. I use it in given up cases. So because I definitely want to utilize the drug, so there is a space for this drug for me, but unfortunately not as a first line drug. So if you have, means fortunately or unfortunately, any kind of incidents, these are the protocols that were given by our group and by Baumal group, so that you can refer. This is again our proposal. From our learning from biosimilars, we propose that BioBetter could be something which can change brolucizumab. Now I'll tell you that why Novartis is not going through the biosimilar route, because BioBetter route is not clear in the world. BioBetter, if you want to design, means what? As a naive understanding, an understanding of biologics, I feel probably, industry leaders are there. AIOC is sitting there, has a great understanding. So, means what I feel is, if you can reduce some kind of molar concentration in this molecule, there is a possibility. You might compromise on some kind of efficacy, but there is a possibility that molecule will survive. Unfortunately, FDA and EMA, they do not have a clear pathway for bio-betters. They consider them as an innovator molecule, and they have to go through the entire process all over again. So nobody wants to do that. So if you look at realistically, PDS, port delivery system is a bio-better. But has it been, known as a bio-better? No. It has been known as a new drug. But if you look at the port delivery system, it has a better delivery mechanism. It has a little, different concentration of means ranibizumab that they use in. Similarly, if you just look at Lucentis was essentially a bio-better. It was a bio-better of bevacizumab. It had just the FAB portion. They just cut down the FC portion. So it means if you have some mechanism by the FDA and EMA that bio-better can have some abbreviated route, drugs like this would not die, because these very important drug. So I still say the brolucizumab story is not yet finished. A wonderful drug, I use it as a third-line drug. So probably we'll learn more and more. So I'll keep it short here now, and because we have two legends sitting with us, so I think discussion would be more fruitful with them rather than me speaking. Thank you so much.
Very good, very nice. Great talk, Dr. Ashish. I would like to just share, I think when all these things were reported, we didn't use brolucizumab. And what Ashish mentioned as in cases where other drugs didn't work at all, that also restricted to AMD, because that is where their approval is in this country. So we did use, but avoided all one-eyed patients, where only one eye. And I would like to mention here we've been the one who has seen one case of vasculitis. We've just, I think, sent my colleagues for publication, but the patient was lucky. 2 injections were given somewhere else, and it was during the second injection, about 5th, 6th day, vasculitis developed. I think it restricted itself more to the periphery. The vision was down, but not that much. She still survived. Otherwise, it would do. And another patient I would like to share, he was a senior officer, Vice Admiral, retired from the Air Force. I think they are very liberal in Army hospitals. So they were giving monthly injections, same problem. And this gentleman, he comes that I'm suddenly having a lot of floaters. And he was sent specially to me, and he had a lot of cells. There was no vasculitis, but he did respond to just steroids and was all right, and we advised not to use brolucizumab for this patient. So we have used this as what you said as third line, not in one-eyed patients, only where the treatment-resistant patients are there. It's a great drug. I must say it does work in such cases. But then that is where our confidence lies. But that's not true for the entire nation. We have seen people using it quite liberally also. But let's see. I think that is what we always say. Come out with better data. Use some kind of a system. Are you using it with the losing dose, 2 monthly, 3 monthly, or simply right from beginning PRN? Because we have to understand, how the drug has to be used, and especially all population also behave differently sometimes. So that can also be there. So these are some of my observations.
Dr. Natarajan, please.
And I think that brings to a good homework for Ashish, because Ashish is involved deeply, and I'm very happy this part of the world is respected for his experience, and they are really looking for. So I think we should have a preferred practice pattern for using the biosimilar, and as you said, also more define the bio-better on what is it and how, as rightly Mangat mentioned, that I think people don't know. They just keep on giving injection, and patient keeps asking 10 questions. They want to say, okay, change the drug. So I think with all my experience, I recently, I won't say burned my finger, but a patient was refractory, but the vision did not go down, and I was using aflibercept And I don't know, I asked Dr. Jaydeep, my colleague, and somehow he likes the brolucizumab, but I forgot to ask him question, what is that protocol they are using? Because I went to the Singapore meeting where this guy who developed brolucizumab gave the talk. And then and that time he mentioned in the beginning there were a lot of problem with the drug, and then later they were refined and all that. But still, and when I sent this report to Neil Bressler, he immediately wrote back, which I mean, he said, we never use brolucizumab because we are not happy and we are not, it's not safe. So that, as he rightly said, we have a group of doctors. I know the company is pushing. This is a problem with, I mean, when I ask the same question, there is a reluctance for me that I don't want to give, and only when it doesn't work, and as he says, in a one-eyed, definitely no. And then the other one, when you have a refractory, why not we have a definition? What
Sure.
is the preferred practice pattern? Okay, they can choose any drug, but when particularly choosing brolucizumab, and maybe they claim 1 out of 10,000 vasculitis. I don't know what's the incident.
Yeah,
But
means
still
1 in 200 probably. Means that's
1 in
what they-
200. I mean, 1 in 200 is very. I mean, all of us will have that immediately because every patient we are treating. So I think that scare is there because how can you do it on your own father? Will I do it? I don't do it. And I ask the company the same. And
True.
I consider every patient the same. Touch wood, the day I gave the injection, I wrote to him and he said, we never do it because of this vasculitis problem. And I have seen reports, fortunate, I have not seen any patient having, and even with any of the drugs, razumab or the other ones. And I always hesitate because when the patient says, I can pay, why do you want to give a, they don't use the word cheaper, why do you want to use?
He is a VIP consultant, so he gets all the patients who can definitely pay.
No, when Visudyne came, Dr. Tiwari said, I'm only doing, in VRSA, every time we'll fight, oh, you're only giving a 1 lakh, because that was 1 lakh including the tax and other things, 1,02,000 I remember. But I introduced PDT first, and like it proved it's a thing, and not just because it's expensive. And I think I'm glad our own home minister got it, and I think he got the price down that time. So that's why I think we need somebody to work on it. And I mean, every company, all of us want profit, but we are not working only for profit. And I think that I like that word from America, that not for profit. What does that mean?
Although they are all for profit.
No. The meaning
true.
is you make profit and
True,
put
true,
it
true.
back. Okay,
Yeah.
fine. Same thing all of us
true.
doing,
Yeah,
and
correct.
I think the company also should do.
So, Dr. Dogra, means in the interest of audience, means I would like you to clarify that what kind of differentiation that means as a clinical differentiation that they should see, that and when they should see in any of these biologics, and what should they see clinically?
You see, for me, if you are really using a drug initially, the most important part is a very closed follow-up. That is the most important, because we have to be very careful. If something is happening, you are not going to call that patient after a month, as we usually do with many of our patients where we inject. And so that is the most important thing. And even in any patient where we use brolucizumab, we call them back in fact very next day, and then after 1 week, because we are scared. You see, so that is never happens with other drugs. It is not happening with razumab, absolutely not. And in fact, our population is such, a lot of patients, as those who can't afford, they have gone on to razumab. They are very happy. It acts very well, and I must say, congratulate that way the company for now having a very stable product, which in fact initially we had big hiccups and we were not using. So I think that is something very, very important. And secondly, I think when we start using it, I think one should be always careful explaining everything to the patient. You see, everything should be written down. We should not be just taking anything lightly. For a drug like brolucizumab, we have a special thing.
Consent form.
Yes. So because I think we are still, and we are hardly using, very, very use is very sparing, I must say. But that is what when the whole world is saying. You see, even one case, if they are going to give in a one-eyed patient, and with this much of a literature coming from West, and you have already published quite a bit, I think it is going to be a problem. Nobody would save the skin of a person who is going to be absolutely hanged. It can be a serious issue. So I think we have to take it seriously.
Yeah, so means as according to sir means that the main thing is you just go slow and means careful examination follow up. Nilesh means I have a question from you means Nilesh means he was my student, understands basics of eye very well. So I have a question from him. Why do you think I did not tolerate this?
Yeah. So as I mentioned, the brolucizumab had 2 or 3 major issues. There was a high molar concentration that was happening. That thing, so much of doses being delivered so near to the retina and choroid, and it was all taken up. The penetration was also 25 times higher than the Lucentis, and the concentration itself was 12 times higher. So it is a large dose we are giving. And then there was pre-existing antibodies itself that was disregarded. It was not taken into consideration. It is a small molecule, and there is a molecular mimicry that is happening. And pre-existing, there is around 50% ADA rate was there. And then there was treatment emergent ADAs also. Like once you inject, you are getting more ADAs. So much of antibodies antigen you are injecting into an eye, and there is a pre-existing antibody. So immediately there is going to be a immune complex formation, and the complement deposition can happen. So that's what we used to discuss. But the thing is, it is very difficult to get implemented. Is either to reduce the doses or to induce some kind of thing where this antibody antigen that bonding can stop. We can add a pegtail or something we can add in the molecule to make it less immunogenic.
So I forgot one thing which is very important. In case even you see iota of little inflammation in an eye, I think don't inject it there, even if little bit, because mostly we are not used to looking at for little inflammation before injection.
I agree
I
very
think that
much,
part
yes.
I forgot. So that's how, Nilesh, what you brought out, so this came to my mind. This is very important. We keep emphasizing that examine carefully. There should not be any even iota of inflammation or cells there, and then don't inject in such a eye, because that could trigger what you have been already sort of establishing.
So probably, that sir's point is bang on correct. Probably, sir, means we have had a meta-analysis means which has come for revision. So because now Visudyne is not there, PDT is not there, and if you see the Japanese data, the polyp closure is around 80% to 90%, which was not even achieved by PDT. That kind of closure rate brolucizumab provides. But meta-analysis, if you see all those, right now you have just 5 reports. Out of 5, 4 are from Japan. And out of those 4, you have around 11% to 15% cases with inflammation. Indian report is 1, 0 inflammation. So that comes to your point that probably, we as a clinician or we as a country means the same thing what happened in Hawk and Harrier, when the IOI was there, periphery was not examined well. So probably that's what we missed. That is why our incidence, although means we are not losing eyes, patients are not blind, but as you said, tiny bit of inflammation, probably that's where we are not able to detect. Now means we have representation from Novartis, so means we would like to hear from you also, means Novartis, means if you can just put on your points, because means this we have made it as a unplugged session, so because everybody can participate, means if you can. Yes, is there, right? Alok is now means I have learned so many things from Alok Russell. He's sitting there. So because he has been in the journey of Razumab since the beginning, and means even in a lot of our biosimilar work, I consult with him that this is the kind of thing, I have this kind of presentation. Means he's involved in right now EM and FDA trial. Alok, I have your comment on the phase 3 trial design, which we are also a part of. How this is different and why people should have confidence in this trial, in this design.
Thank you, Dr. Ashish, for this opportunity. To talk about trial in front of 3 of you is something which I could have dreamt of. How the phase 3, we are getting into phase 3 now, so you are center. PGI Chandigarh is center of our phase 3 trial. What we've done differently in the phase 3 trial is have different races typically, which earlier used to be only US and Europe. We've added a Asian variety, which includes Indian subcontinent and Europe. And typically FDA response was, we are fine as long as you have any light-eyed and white-skinned patients. So that's the variation which you referred to as Japan and India incidence change, which does happen. Second is for this typical trial, though the protocol mandates certain inclusion-exclusion criteria, we have kept a central reading center which has very bigger span to study the images. So typically like the peripheral parts which are generally not seen, or we typically focus in trial on only primary and secondary endpoints. Our secondary endpoints is a list of endpoints which probably would lead us to a better understanding other than just the regulatory primary as well as the secondary endpoints. And like you know, sir, the inclusion and exclusion criteria becomes very, very significant.
Tight.
Because that's how we get a homogeneous population. Otherwise, then it gets into all sorts of subgroup, and finally we end up with a lot more questions from a trial than answer. So
Raja, you can come here.
That's it from
Oh,
my
Raja
side.
is there?
Yeah.
Oh, Jay is Jay.
Oh, he's not right.
So bang on correct. So means if you look at means why I personally, because this is the question that everybody has. When I was in US, the question was, people had disbelief on the trial. So means I would like to emphasize here that trial phase 3 means we had an invitation from Dr. Means FDA, who means reviews all these trial designs. So means we had a paper, we submitted, we got rejected, but we means fortunately we were contacted by the FDA reviewer directly that Ashish, I didn't wanted to reject this paper because the phase 3 trial of biosimilar has to come in the lights of people so that they understand that why this design is. So then we wrote again, and the editorial is published in Ophthalmology. So I'll just summarize it, that why the design is different. If you look at the, normal biologic design is a normal pyramid. Okay, and so pyramid means at the base you have all the pharmacoequivalence data. So for example, I'm just opposite. So biosimilar design is normal pyramid. So basically at the base what you have is all the pharmacoequivalence data, and when at the apex you have the clinical data. So apex is small. However, if you look at the reference molecule means which is being approved by FDA and EMA long back, that is this kind of pyramid. So at apex is down. So apex is down means you have less analytical data, but you have a plethora of clinical data. Why is that? Because you are working with a molecules which has never been used, and you have no idea how this is going to behave in human population. That is why you collect plethora of phase 3 data, and that's where the difference comes. You need to have 2 studies for phase 3 for FDA and EMA as far as the reference molecule is concerned. And that 2 different studies for different indications. However, when you have a biosimilar, you already have a drug for which safety and efficacy has already been established. So you need more of equivalence data, and it's like kind of a stepwise approach. You give that data, then you come, then it's an animal data if it is required, then you come to just find whether biosimilarity has been completely established and we are not missing any clinical difference which is clinically meaningful. So that is why you need a small data, 1 trial, and you have an extrapolation of indications. You get 1 approval, you get approval for everything. So there is a sense in this design, and the testament comes from Europe. If you look at Europe, Europe is the most matured nation as far as biosimilars are concerned. Europe has approved its first biosimilar in 2006. And today we are sitting after more than 15 years. there has not been even a single biosimilar across the Europe and across the globe which has been withdrawn, which has been suspended even. So that is the kind of regulatory, strength is. So that's what I always say. Even in US, in the session, I said, you look at the kind of, so believe in your regulatory, that how the regulatory are designed. There is a purpose, there is a sense that they design that kind of protocol. But as Dr. Mangat said, means any new drug which comes in, we always need to take it slow. So, sir, you have any other comments?
Most of the things have been covered, and I have already spoken about the way we are. Of course, we are looking forward to if it gets, we get this drug in a better way somehow when we know it, whether you use it 2 monthly, 3 monthly, and you don't get it. So that part we are just wondering. So but we are not, we are using just as PRNS. That is what I'm saying.
Sir, I had one question to all three of you. For the one like me who is just into the practice, the how to go about choosing, because we have now 2 molecules that are having similar efficacy, similar safety, and if we take for Razumab, the Ranizurel and Lupin, they are still in naive phase. So Razumab, I have Lucentis, and I have to counsel the patient which to take. So he asked me which is better, and then it comes to only finance. So is there a logical answer to it, when to choose biosimilar and when to choose biologic, or the innovator, or should we only go on finance if the patient is capable to pay? We are going to go for.
I think that's what I said in the beginning. For us, is building that experience with both the drugs, and you have used enough. In fact, the way we are doing it today, we don't write now at this time. It is Lucentis or Razumab. We write Ranibizumab. If it is mostly AMD, we write monthly for 3 months. Usually we don't write that for DME. We don't believe in that kind of theory, monthly only 3 injections. There it could be sometimes 5, 6 injections, more injections, right, or monthly also. So we do. So what I'm saying is, it doesn't matter. Sometimes they do have their own inquiry, and they would like to, why there is a difference of cost and all that. So we do tell them that this is the drug which was primarily, I mean, they also know that that one drug which is coming from outside, and probably it costs more, and so it is sometimes something like using so many products. It could be even IOLs, other things. It boils down to the same thing. So we don't really enter into that kind of a thing. So that is what we have done with the, as far as the Lucentis and Razumab are concerned. Yes.
So I think here means, to follow up your question, Nilesh, brilliant question. So Dr. Natarajan, means now as the finance comes into play, means we have seen West. What is on top? Eylea. If you look at kind of usage Eylea has across the globe, means we are completely opposite. Means we do not have Eylea users, but if you look at now their pricing, that comes down to almost like Lucentis pricing. With their
that is what I would like to say. If you ask me now, the number 1 anti-VEGF used in our center is Eylea.
Absolutely.
After the scheme came in, because it
means
comes
our
to
usage
it
also
is
become
pricing,
high.
and it is more effective, no doubt. I must say at this time, majority of our patients are on Eylea.
Happy with that.
Yes, and they are very happy. You see, and we can even increase their duration down the line, and they need lesser injections, and it is more efficacious, obviously, because of the way the product itself is.
True.
So it has changed after the scheme.
After the scheme, yes.
Yes.
No, I know. From the beginning, I always told that if you can afford, I take a Aflibercept. Otherwise, go to the Lucentis. So I think my patient chose Aflibercept from the beginning, even before the rate. But I agree with you. So I think finally, I think the question to answer to Nilesh is, I think the data. So I think we have to need a big list of patients also you are asking. So you can say how many thousand patients have been injected with whatever the Lucentis and Aflibercept and Ranibizumab, and what are the problems faced, and then you choose. And the problem is we need a meta-analysis from Ashish to see. I mean, I don't know, very rare patients ask us this question. I think the patients in India totally believe you. Whatever you say, they will do. I'm not saying they don't believe you like God, but at least they have a belief in this matter because they can't analyze like Ashish. Or even, no, I'm not saying, seriously. See, I think they will compare cost, they compare vision. But I think when you say AIOC2022 is the best and it's more expensive that time, and then Lucent is the next, and then it's good, and there's so much cost. So some patients choose Lucent, but my patients always selected. In Bombay, it's a problem when you are charged the most, and that's the best. And unfortunately, the Zara, the Gucci, all are there. So they are all deal with it. And I think the question, as a scientist and as a clinician scientist, I think it's our duty. Right. What will you do, and what's the scientific data we have? And the answer to the patient when they say the cost, I said, this is better than this, and this because there's so many thousands have been done, and problems are only this much.
Brahm Prakash, so means you should come and speak, but because I have seen West, means Eylea being used, so you should tell me to the attendees that what kind of pricing is down now, because that's the point which Bayer has never highlighted for such a wonderful drug, and the usage has not gone up the way it should have gone up. So just for the interest of audience, now Eylea, which is a leading molecule globally, has a price almost equal to Lucentis, and sometimes now the results of the trial which is going on, where they do kind of high dose Eylea. So although it has failed in the primary endpoint, but still you can have approximately 2 to 3 months kind of dosing. So that result we will have very soon. So
hi everyone. Actually, we have launched this Eylea for You program just before the lockdown, and the program is as follows: like patient is going to take first injection, second injection comes free to the patient. Patient is going to buy third, and fourth or fifth injection is going to come free.
So what is the cost per injection comes to clinic as of now?
Yeah, so initially, sir, once we launched, and that time price was approximately 56,000. Still it is the same, but with the help of the Eylea for You program, if you see the cost of the injection, that is coming to close to 22,000 rupees for the patients.
So 21,000 is the pricing. So it matches approximately Lucentis pricing.
It is even less than that.
So you should highlight it more, because a lot of people wants to use Eylea, but because from the beginning it has gone 60,000 in my head. So it's just gone in the head. That is a costly drug.
I would like to mention here, you see, it is very interesting. In past, I wanted to use in diabetic patients especially, injections maybe for 5 or 6 injections monthly. It will never happen. Now with Eylea, it is automatically happening.
Yeah.
Because it is...
True. Like a very interesting point. Very interesting point.
The third injection, 2 are free. So they are ready.
Yes.
2 injections are there. And I must say, I have shown some cases. And if you do give monthly 5 injections of Eylea at this time, I think your results visually, fluid-wise, as well as later on the number of injection required goes down drastically. And that is what is being propagated from the literature in the West.
True.
That is what we used to always listen whenever we listened to the Neil Bressler's talk or other people's talk on DME. And that is true, but we could never prove it. Earlier, I never had even one patient who had received 5 Eyleas monthly. Now we have several. So I must mention that point also.
There is one more point which we want to highlight is that because of this program, the compliance part has been taken care by the patient very well.
That's what sir said.
Patients are taking those 5 injections.
No, sir has actually pointed out so well that even in my practice, PGI is a big institute, but even I do not have a large practice, but it still means that compliance, that people take 5 injections because they come. This is my free injection. Now after 2, this is my 3rd, 4th free injection.
Yeah, that's a
so that
typical
actually
American.
That becomes a monthly injection dosing for them.
American shopping mall culture where buy one get one free, and then they say it starts from $5 to whatever. So that $5 is never available. But when you go there, at least because of that you go into that shop. The same thing is happening with the people's mentality, because they don't understand science here. They understand the economics. And I say correct. And I think that's why I said it's our duty to, as a clinician,
Yeah,
scientist.
I would blame Bayer for that as of now, because because that has not been put on any podium, because that such a wonderful drug which rules West, but if you look at our data, means whatever we have, although we are poor in data as a country, but still our users of Eylea are less.
Pradeep is not here. I think so much I pushed Pradeep from Bayer to talk. And I think one of the first.
Yesterday we had a meeting. Then I told this should go. This should go to the people's mind that this is not a 60,000 drug. This is a cheaper drug now.
We can say we have financial interest and talk to you.
Jai is a fantastic. He was our DNB student, doing very well. Jai.
Yeah, he's doing excellent.
So I think my question is to all the panelists, especially to Ashish sir, because you extensively studied the regulatory panel, throughout the globally. So there are certain decisions that need more clarification on regulatory and scientific way. So like there's a molecule, ranibizumab biosimilar, that has got few immunogenic reactions in the past. Maybe there have been 2 or 3 cases, and that gets banned by the regulatory board of India. And there is another molecule on the other end of spectrum, like abrolucizumab, that has got a far superior rate of immunogenic reactions with retinal vasculitis and all, but still that gets a regulatory approval and doesn't get banned. So how do you make a sense of this decision?
Very, very interesting question, Jay. So that's a $1 million question. What happens, we look at FDA and EMA all the time. Okay? So if FDA has passed it, so our regulatory takes a call from there. So that is how we have been doing it. So probably, even you are referring to the Avastin case. Avastin was banned, and
Avastin
then because of the effort of everybody, it came back. So it's a weak molecule as far as that, pushing from the industry is concerned. But other molecule is strong as far the industry is concerned. So our regulatory looks at the West regulatory. That is, that's how it has been going.
Yeah, even Razumab was banned for a short period.
Yeah,
after
true.
a couple of
I think that
reactions.
was probably a good decision from the company. They themselves withheld it, if I am right. Correct? You themselves withheld those batches. So I think that was not a ban from VRSI. Correct? It was not a ban from VRSI or any regulatory. It was a responsible decision from the company that they just stopped those batches. But I agree. So we look at West, and that's how the decision is being taken.
Some of us have to get into this advocacy and get into this regulatory, which is difficult. I know, because the bureaucrats are different, and then this regulatory affairs, even in India, even though they follow West, there has to be something we have to use our driving. Now
Sir, this
they are looking
is
at
the
India.
for ophthalmology and retina specialist, and now if you talk about biosimilars, regulatory part is so complex. We had some meetings with Canada. Now, if you look at our paper, which was published in BGO on regulatory, that is the only paper on regulatory for biosimilars. Canada has kept it as a policy document in their government decision. Although, that paper, it was so difficult for us to publish because there was nothing available for ophthalmology. So we had taken all the regulatory, what was there for the systemic diseases. But that has become an industry, policy document now for Canada. On the basis of that, now BioVis is approved in Canada. So as you are rightly saying, some of us probably has some kind of involvement there.
Yeah. No, but they also don't want. I know that.
What is very important, Ashish, if you have this paper, you see, for anything in this country, you need to bring that paper to our regulatory. It will be there in Canada, but not with us. I may tell you, I worked in a very difficult area. Dr. Natarajan knows ROP. When we had to change the guideline, they always again look at the West.
Yes.
It's very hard, very difficult. They still had 1,500 and those 30 weeks or whatever. We wanted it 2 kgs and 34 weeks. You see, it is only, even if the paper would be published in an Indian journal. My student was Anand Vinekar. I gave thesis to him. That paper has more than 150 citations from Indian journal.
That's right.
Maybe
He deserves a big round of applause for changing that.
No, what I'm saying is why I brought this not because of any other reason. Like
I remember.
We got that paper into this thing, and it went to all the people, and they realized it. In India, things are different. These babies are going blind. You see, higher birth weight. So what I'm trying to say here is the same way.
True.
If your work is being taken seriously somewhere and it's changing, it should come also with our regulatory people,
Yeah, I
those
think
who
we
matter.
So
should
you have
do-
to help
Yes.
us, sir, in
No,
that.
no, we
Yes.
should do publication in IO with this, keeping India in mind, and also push, with the health ministry, with the drug control of authority of India. We have to, because once upon a time my patient was a drug controller. When I went, the typical government attitude, whatever you say, like I remember there was a application that time, one of the Parsi person was telling that we should ban that diclofenac because you know why? The vultures were dying.
Ah.
And then they couldn't bring anything because they said, why are we bothered? But he's saying, no, diclofenac is killing the vultures and it's becoming extinct because
So
they're taking so
probably,
much
yeah.
diclofenac.
So probably, sir, actually it's very well pointed out. So probably what we would do is, because now even your center, our center means we are part of phase 3 trial for aflibercept.
Yes.
So probably, before aflibercept comes in, probably at least, because I am seeing, even inclusion, exclusion criteria is quite varied by different companies. So probably means if we have something streamlined,
Yes.
and then we put it to the regulatory by publishing here, I think that would.
We've done a lot of work in this
So-
area. It should be brought out. I mean, if it's just for the
A lot of homework
people,
for it.
yes,
No,
yes.
really,
Homework,
we
yeah.
are there. I think VRSI, we have now a vibrant again. For current 10 years, I think they are doing a lot of things in par. And now I think rest of the world is looking at, because of you and Barry Cooperman and Francesco, I think people are looking at Ashish as an important person, which is important for us. I think same thing can be encashed here. And as he said, we have to publish in relation to drug control of India and find a way. And we are not
So that you have to help us in finding a way to get connected.
100%. No, we'll do that.
Yeah.
We have to do that. We have to keep pushing. It is like a rock, and we have to break some of us have to knock. He did that for RFI, I know that.
Yeah.
So similar thing we have
Even
to do.
the time when Avestin was what I think, because of all your
Yes.
help, I think that is.
Yeah,
that is-
we
that
had
is
a
brought
problem
in.
because even in France, the Avastin, the government told Roche that if you do this, we will debar you from other products, and that's how they came back to in Roche. They accepted, and then otherwise in India they gave a certificate saying that it's not for ophthalmic use. We had a tough time to
In
fight
fact,
with
it
them.
was banned when I was still there.
Yes.
I was.
Yeah, I remember
I was
that.
the chief of retina, and it happened with so many patients. Of course, we could save all patients, but it was banned because of our institution use, and it was one-day use.
Yeah.
And you see, then they wouldn't agree. They took all our Avastin, something like 28 vials. And not only that, I still remember the whole group which came, they threatened me that you being a chief, you have done two things. One, you have used a drug which is not used for humans. And secondly, no, not for the eyes, which is meant for other use. And secondly, you have used off-label drug. Even off-label drug. I mean, I said off-label, so many drugs are used. But that is what the way they behave. They can behave very
nasty.
This is a
Very
drug
nasty.
controller team.
So suddenly they get power,
Yes.
and they want to show the power. That's the problem.
And till today, I am now superannuated from my institution. PGI has not used even a single Avastin after that. Can
I
you
have never
imagine?
used.
So that's-
In my private practice, I have never used. Only the charitable place we use. That also the others I don't use at all because of the same problem. From the beginning, because every patient expects a great, and even 1 out of 1,000, they don't accept it. Like a,
Yeah, Alok, please.
Sorry, but I would add here something is typically regulators would see it in a fashion which is more safety driven. They do not spend more time on efficacy or economics of the disease. But what we see now in current change in regulatory is they are now becoming more sensitive. They are at least adaptable or willing to listen. And the first thing is by establishing those secondary groups or the specialty driven groups. Typically coming back to ophthalmology, I'm not going into the bigger areas still today of cardiovasculars, where there's a huge, enormous patient as well as economic load on the country, oncology. But I come back even to ophthalmology. Believe me, DCGI has difficulties in getting those expert groups together. We always say VRSI is active, we always say AIOS is active, but when it comes to DCGI's meeting as expert committee meeting, I attended 3 of them and none of them had a retinologist in them.
Yeah, I know. I struggled when I was there, tried to get in. I couldn't get into that in spite of knowing some minister.
No, people don't attend it, sir. The problem is different.
Okay.
Because they will meet on that day, which is usually communicated 3-4 days before, typically government fashion. But we do not have experts attending those meetings.
So how can we bridge that gap?
One thing probably which I don't see this group discussing is how can we actually partner with the regulators more.
Yeah, that's what I'm asking.
So I think you have to let us.
And typically it's usually the RP center head, it's typically Saftarjung head, Lady Harding head, which are typically Delhi driven because of their accessibility and their mandate to attend, and they are not, forget ROP, sir. We presented for ROP. There was not a single pediatric ophthalmologist there.
No, I know. I
forget
think,
not retinologist.
you can help us unofficially to say, or at least to me, and I can convey there and talk to the VRSA also, and as a part of AIOC. That is what I tried doing, liaison between AIOS and government. Somehow nobody came forward in the governing council. Everybody wants to do. See, in US, I kept on telling there is a group called Surgery by Surgeons, where they only talk 600 ophthalmologists from the academy, have a separate meeting in that hotel, I forget the name, and they don't come to the academy at all. And they have a meeting only on this advocacy, going to government, because there they have 50 states have different problems in the same issue, like right from optometry to some drug license to practicing license. So I think we should start that in
this bridge
but
has to be very well brought by Alok. So this we have to bridge.
No, what I'm saying, Ashish, I want to use the word retired people like me and Mangat, but not, I think that's not the correct, because the government will not accept that. But we should have an active person from the VRSI. At least for retina we can talk. With ophthalmology, I tried my best for 10 years, nothing happening in AOS. But I think VRSI, I can talk to Mahesh and Muralidhar and say, me, Mangat, and whoever like Cyrus from Delhi, I say that we have to represent and say we need our role in the DCGI, and then as he says, the expert committee and other ones. See, I remember ICMR has got that for retinoblastoma, and that is because oncology is very important for them. Rest of the thing not there. Even in diabetes, I'm trying to meet the minister and do something. And I think for the drug, and because India is going to be the leader according to me, whatever I'm seeing in the world, in Europe and US, they have to learn from us.
Sir, my humble suggestion will be VRSI to publish a white paper that can be widely circulated, and because we can't have the access to DGCI, as sir is saying that it is just 3 to 4 days.
We can. What I'm saying,
yes, sir.
By going back again and again, I have learned a lesson where we need dedicated, separate team, not the office bearers.
Yes, sir.
See, when I was an office bearer, it was a problem, because by the time you do this, something else. But we had to have one team where you do only this. Now tell me why Ashish is able to get a name, only because he's working only on the biosimilars and the anti-VEGF overall from beginning. From the 2008, I think, you're with Barry Cooperman, and they are also all the time on the limelight, not just because of the name, no, they are because of the work. So similar thing we can do with a VRSI, making a separate cell for advocacy on drugs. And I think we'll take Alok also as you rightly said, unplugged. We should have people from like the MD pharmacology from the
purpose of this session was, particularly I spoke to all the companies because my experience in Miami was fabulous.
Yes.
We had an opening session with the industry leaders, with the retina
industry
people.
leaders were on the dais with them.
With
us. I
us.
think only two retina specialists, and rest of them were from industry.
Yeah, me had
and the entire audience were listening, because I think that's the first time we saw, at least there's an example that there's nothing like bias that just because the company is giving somebody as a consultant. No, they write everything, but still, I think they were asking the basic questions, that molecule, and otherwise we only talk about pricing and this one, all that, but that's a commercial part. But they talked science with the industrialists, which is why I think we have to start. I'm happy you have done that as an instruction course, but in VRSA Nagpur, maybe they can have a session like that.
Like an unplugged session we should have with industry, because now you see we get so much of information which we can get a lot of help from industry in bridging this gap.
No, Ashish, there is a big chance for you to be the leader, India to be the leader with you as a biosimilar, because I don't think America, even though we discussed with that company, they have got a FDA, going to get FDA approved. I don't know how Genentech, and I'm just openly telling, they may not allow.
True. So
I mean-
we don't expect in US, though Samsung has got that FDA approval, it
Yes.
usually takes year to 1.5 year from approval till launch.
No, now launch is there. I mean, I got an email yesterday itself.
They're saying this year.
No, not this year. Drug is being kept on the shelf
Super.
for use from this month.
Fantastic.
I got an email. I have a virtual call on 20th of June with the
Wow.
Biogen. So it's already available. There even prescription information has come to me. So except DME, they have approval for everything in US. Europe approval for everything. US because of the dosing 0.3 and 0.5. So that they would bring in. The problem is where they would place it. That is a question for them. How payers are going to do it.
Exactly.
And the major part is on-label bevacizumab. If on-label bevacizumab get approved, then it's a different ball game. Then entire US cannot use off-label Avastin
Correct.
because as per the law.
Correct.
So then the care is not cheap. Then the care goes costly because on-label bevacizumab cannot be cost like $50, $100 that off-label bevacizumab is. So it means it's a kind of a regional issues.
But they are calling that as a new drug, right?
It's an innovator molecule. Now my question, my question
They are
is,
calling it like that.
so innovator molecule, why? Because to call any drug biosimilar, you have to have a reference biosimilar.
No, so
Reference or a reference
Yeah, there was a
molecule
big
was
debate
never
there.
there.
No, there was a big debate there.
We faced the same thing. We were, as Intas, we were following up with DCGI for bevacizumab biosimilar, which we already have. But exactly, DCGI, two points which didn't go in favor and we had to drop that development was, one, the referral 5 countries, what are they doing? They have no bevacizumab for ophthalmology use. And second was they said, well, you do a trial from, physical part was good for us, but they said, give us animal data and phase 2 data. Now the moment we go to put phase 2 and phase 3 costs in the product, I'll never be able to market it
So
at the
that's true,
price
that's
which
true.
it is.
That's
available.
probably going to
So-
happen, means with Outlook.
And exactly, Outlook has said $500. If I remember last, what I read was they're looking at $500.
Argentina, if you know, Argentina
that's,
has one on-label bevacizumab.
But nobody is using it.
No, means the
interestingly.
users are less because it's priced approximately $400.
Absolutely.
So-
True. And that's the reason why nobody, even in India, is looking at bevacizumab to get approval.
Ashish, I think since the session is coming to end, what is the take-home message for us sitting in the dais? That means homework. The homework
So I
is
think-
I
I
don't,
think
I don't
means,
taking the homework from licensing with the government, which right now I have time, so I can, there's no post for me.
So I think homework, we will conclude the session in the way means that,
Homework for us.
means with your help only, sir, we will build up. So probably means we have already
Yes,
established
concluding.
a real world group globally, but I think let us come back to India. So then probably with the help of industry, means I'm glad that means we had this session with industry today. With the help of industry, let's get connected with the regulatory. I think that is the one
Yes.
thing.
Yes.
And let's get connected with the regulatory, creep in there with the help of you all because
Yeah.
you have that experience. And then regulatory wise definition, defining, that we would do.
Okay.
That how we should define. So let us have a proper protocol wise so that all the biosimilars and whatever is coming, at least an India-centric protocol should be same.
Okay.
It
Good.
should not differentiate.
So why
it.
not we publish that? And
Yes,
go
sir.
back to the company.
Yes, sir.
again.
We will surely do that.
We'll do that. We'll work with it. So I promise publicly.
So thank you so much, and I think I should say a big thanks to Dr. Mangat and Dr. Natarajan, because we were thinking that we are just two, which are going to handle this session, but it turned out to be a brilliant session.
No.
Thank you so much.
2 is equal to India, and India is equal to 1/7 of the world. No, really. No, I'm proud of what.
Nilesh.